During early development, a very large single-celled egg is transformed by successive cycles of cell division into an embryo of thousands or millions of cells. These three eggs were fertilized at the same time and started dividing within minutes of each other. The events you are seeing last many hours. They have been compressed into a few seconds by time-lapse photography. We are viewing the egg from the top. This surface is called the animal pole. It is dark because it contains a layer of dense pigment particles just inside the egg membrane.
The embryo stayed the same, but it consists of a whole lot.
Once the embryos consist of some thousands of cells, changes in their surface tissue create a ridge that will eventually turn into the backbone of the tadpole. Next, the tadpoles develop a kink on one side, and spontaneous movements start about 28 hours after fertilization. The embryos elongate until they rupture the egg membrane and escape about 50 hours post-fertilization. As the embryo grows, its eyes and tail develop while its internal organs take shape. The gut cavity contains the dense yellow egg yolk, and it shrinks steadily as these nutrients are used up. The embryo acquires patterns of pigmented cells called chromatophores in its skin, and by now it is clearly recognizable as a tadpole. For convenience, we will now divide this continuous developmental sequence into 5 arbitrary phases and look at each in more detail. Our first phase covers the early cycles of cell divisions, starting with the single-celled egg.
That's cleavage going on.
Our second phase continues with a vital process known as gastrulation, which precedes differentiation of organs within the
outside
embryo.
of the embryo. You can't see the inside like you do on the model. That's the yolk plug, by the way, that you're supposed to find.
Our third phase is called neurulation, when the future backbone starts to form. Our fourth phase covers further elongation of the embryo and its escape from the egg membrane. And finally, our fifth phase shows some of the events of later differentiation as the tadpole becomes more recognizable. In side view, the egg is slightly flattened. About 90 minutes after fertilization, the first 2 divisions take place.
You see where the
The upper layer of dense granules marks the animal pole. The lower part of the egg, where the yolk reserves are concentrated, is called the vegetal pole. As the cycles of division continue, the animal and vegetal poles remain distinct. Gastrulation takes place at the vegetal pole, during which the pigmented cells of the animal pole are pulled downwards. This animation shows the animal and vegetal poles of the egg after it has gone through many rounds of division. Next, we look down vertically at the animal pole of the egg. The first 12 to 13 cell divisions are synchronous and about 30 to 45 minutes apart. Meanwhile, the cells start to separate internally, creating a central space called the blastocele, not visible from outside. Continuing cell divisions transform the single layer of cells into 2 layers. Soon, the egg has become a partly hollow sphere called the blastula. Viewed at high magnification, the surface of the egg puckers briefly as it is constricted during the first cleavage. Thereafter, the layer of pigment reveals the successive cleavages dividing the egg into smaller and smaller cells.
What's going on is mitosis. This is
We
the
cannot see
cytokinesis
the other
part.
divisions
We
that
learned
create
about
the
that
two
before.
layers of cells.
The animals, they pinch off
Eventually,
like
from
that.
this view,
Cleavage is how
we are
cytokinesis
looking
happens
onto the
in animal
root
cells.
formed by these two layers over the blastocele. Eggs are sensitive to gravity and always have their animal pole facing up. These 11 eggs were turned upside down just before video recording started. Within 15 minutes, rotation of the cytoplasm within the egg membrane brings the dark animal pole back to the top before the first cleavages start. This gravity-induced rotation of the egg cytoplasm is now viewed at higher magnification. Just why the cells respond this way to gravity is not known. If this reorganization is not completed in time for the first cleavage, the subsequent cell divisions appear to be unaffected. The next critical event of embryogenesis, gastrulation, takes place at the vegetal pole. This egg is viewed from underneath. After numerous cycles of cell division, gastrulation starts with a marked infolding at one particular region on the lower surface. The activity spreads across the surface, steadily sweeping the outer layers of cells into the blastula.
So it's the veggie pole, huh?
Gastrulation generates several new layers of cells within the embryo, and these layers will soon interact and differentiate into the major internal tissues and organs. This animation summarizes how the complex events of gastrulation create new layers within the embryo.
There's the gut forming, and the blastocele gets smaller as the gut gets bigger and eventually goes away.
The sequence is repeated, and arrows now indicate the inflow of cells that can be detected in living embryos.
That region at the top is called the dorsal lip of the blastophore, and it was determined
Finally, this
a long time ago
simplified
that's an
diagram
organizer.
shows the
Without
new
that,
internal layers of
cells don't
cells,
know what to
the
become.
ectoderm in blue, mesoderm in pink, and endoderm in yellow. From these layers arise most of the important tissues and organs of the tadpole. During gastrulation, a pore called the blastopore is formed which steadily shrinks. The movements that bring about gastrulation are generated by individual cells changing their shape, stretching and contracting in a coordinated wave. This activity causes the surface to flex and then be drawn inwards. Here is another sequence showing these critical events, which are completed after about 14 hours of development. Experiments indicate that the lip of the blastopore acts as a center for organizing many subsequent events of embryogenesis.
Hans Spemann figured that out. He won the Nobel Prize. He was the first embryologist to get the Nobel Prize for that.
As the blastopore closes, it defines the position of the end of the backbone about to be created during the next phase of differentiation, called neurulation. Neurulation starts with the creation of the neural tube, again formed by carefully controlled movements within the embryo's surface layer of cells. These movements form a tube arching over the embryo. Once formed, the tube is raised by a growing ridge called the neural crest. Then the whole embryo suddenly kinks on one side and starts growing lengthwise. Neurulation starts with an infolding developing along the embryo's surface, here seen from on top. Soon this invagination seals tightly along its length. Once the tube is formed, the neural ridge slowly develops, curving around the embryo. Here, the tube is seen in end view as it closes up. In this higher magnification view, many individual cells can be seen along the enfolding surface. Neurulation depends upon the coordinated extension and contraction of these cells, and, finally, their adhesion along the length of the tube to seal it. Here is a final sequence of neurulation. It shows the wave of activity generated in the surface layer of cells. The embryo often rotates within the egg membrane during these events. After neurulation, the blastula undergoes a sudden contraction on one side. This contraction is immediately followed by pronounced kinking of the whole embryo. The embryos now become active, often twitching violently.
They call it tail bud stage, usually, I think. But that's probably good. I'm going to stop right there.
Certain cells on the surface of the embryo are ciliated. This sequence, in real time, shows that the active beating of these cilia maintains